Welcome to our electrophysiology unit that will be a several-part PowerPoint presentation. For the first part, I would like you to be able to identify the history underlying the development of the action potential. I'm about to show you a video here that would just be for enrichment purposes, but after that is sort of more the information that I would be really testing you on in terms of history. I would like you to be able to describe how electrical information is conveyed to cause the spinal reflex. And I would like you to be able to identify how we measure electrical conductance in neurons at the very general level. So if you thought my little history lesson on Ramon Cajal versus Camilo Golgi was interesting, this should blow your socks off. The history of how we found out that the nervous system conducts electricity as a signaling means was really started by Ben Franklin. Ben Franklin, as you might know if you've seen the musical Hamilton, was fundamentally involved in the establishing lightning rods and these sorts of things. But this all started when he had this notion that, lightning was electricity. We could harness this. We could even use lightning rods to protect other things from lightning. We could control it in some way. So he sent off this idea, to an academic journal, in Britain that rejected it on the basis that he lacked any observation of this, like actually being possible. So he went back and performed the famous kite flying experiment and published that finally. And that captured the mind of some scientists in Europe, one including Galvani, who you probably recall from the galvanic skin conductance task. And if you actually look this up, Galvani's wife actually played some role in this as well. But I'll show you what Galvani did here on the next slide. So what Galvani did is he replicated Ben Franklin's kite flying experiment. But what he did differently is he had this idea that, to test the nervous system conducts through electricity. Let's attach a nervous system to this kite and see what happens. And that's exactly what he did. So he took a frog that he first killed, and then he severed the head. Luckily, he didn't sever all of the brain. It actually turns out if he severed the entire brain, his experiment probably would have failed. But he, put the kite up in there during the lightning storm and attached the wire, an electrical conducting wire, to the frog's nervous system. And when the lightning struck the kite, the frog appeared to be reanimated, jumped around, and that is the story of how we first found that the nervous system conducts through electricity. It went from Ben Franklin to Galvani, and then this led to widespread, arts. Like all the artists, their imaginations in Europe was then captured by this series of experiments. One person in particular, Mary Shelley, took note of this and entered a contest with her husband, Lord Byron, to write the most scary horror movie or the horror book. And she wrote Frankenstein based off of inspiration from this experiment, which shouldn't be that hard once you see it. And then, there wasn't TV at the time. It was very common that at that time there would be lots of street performance or street art. And it was very common that you would have these reanimation experiments. It was like what zombies are, or what, some of these other ideas that are more popular today, superheroes, they had reanimated corpses. That's what fascinated them. And it all really came from neuroscience. I mean, you could say that neuroscience led the way here. Of course, a physicist might say that, well, Franklin, with his biophysical ideas, came first. Fair point. But, neuroscience definitely played a role there in paving this idea of the reanimation of the corpse and all this, and science fiction as well. I believe these cartoons reinforce and enrich this content. Ben Franklin's famous experiment harnessing electricity made sparks across the scientific world in 1750. In particular, it charged the curiosity of Italian physiologist Luigi Galvani, who applied an electrical current to dissected frog legs with startling results. He suggested that this animal electricity arose from a property of the tissues that we now know relies on ion channels. Zapping the nerve bypasses the signal from the brain that causes these channels to open, activating the muscle fiber. The ability to cause muscles of the recently deceased to contract led to many shocking popular demonstrations of animated corpses, which then sparked the imagination of an Englishwoman named Mary Shelley, who created the cautionary tale of the infamous monster. And while I have my cartoon book out, I also wanted to throw in another one here, which I think also harkens back to a point that I made in our last unit, or last lecture, which is, every species has every question in science has an ultimate species to get at. So, if you want to measure electrical and chemical mixed synapses, the goldfish is an excellent subject. Much better than a human brain, much better than a rat brain. And it turns out that a lot of the, properties of action potentials that we understand were derived not from the rat or the human as well, but from the squid. So, I also thought it would be highly relevant to go over this history. In 1929, Danish physiologist August Krogh famously said, "For every question in biology, there is an animal in nature that is most convenient to study." For centuries, philosophers and scientists had wondered how our nerves conduct electric impulses. What mechanisms allow nerves to transmit such clear signals that don't decrease in amplitude? Enter anatomist J.Z. Young, who, while traveling in Italy, became, scientifically we presume, very interested in squids. Out of sheer curiosity, he dissected the animals and noticed that its main motor neuron is nearly a millimeter in diameter. This caught the attention of electrophysiologists everywhere, who for years had struggled to study the 1 micron or micrometer diameter nerves of most animals. Working with a large enough specimen, scientists were finally able to figure out that nerve impulses travels as waves of ion exchanges between the inside and outside of the nerve fiber. Special proteins embedded in the fiber act as one-way gates that are triggered in a chain reaction, while others act as pumps, restoring the ion concentration from the next impulse. All of which goes to show you, in science, it pays to follow your curiosity, if not your appetite. So in the next 2 PowerPoint presentations after this one, we'll go into some of the fine detail here, but I think that it's best to start with the big picture of neural communication here before we jump or delve too far into the fine tuned details of the action potential. But neurons, as you know, communicate using electrochemical messages. As you now know, Ramoni Cajal showed that there are unique cells rather than just one electrically connected net. And within a neuron, the message itself, though, is electrical based on ion movement, and that can switch between neurons through gap junctions or electrical synapses. But more typically, between adjacent neurons, the message is usually chemical and conveyed through neurotransmitters. Neurophysiology is the sort of all-encompassing term here for what we're about to begin studying. This would be the generation, transmission, and integration of neural signals. By definition, neurophysiology is the study of the life processes within neurons that use electrical and chemical synapses. The primary driver here would be the action potential, which is a rapid electrical signal that travels along the axon of a neuron. A neurotransmitter would be the chemical message between neurons. And it's also important to note that there's a resting membrane potential that would be altered. At rest, this would be achieved by equilibrium or balance of electrochemical forces. These electrochemical forces are primarily ions. Ions are electrically charged molecules. Anions, like chloride ion or Cl-, are negatively charged, and cations, like sodium, potassium, and calcium, which are what you see here, are positively charged. Ions are dissolved in intracellular and extracellular fluid, which are separated by the cell membrane, which results in what's known as capacitance or provides capacitance. So let's think about a big picture concept here first. Touching something hot. Now let's think about this first in just a simple spinal reflex where we're not trying to prevent ourselves from not letting go of the hot thing. So, we touch a hot iron here, by example, and we very much, do not want any sort of contact with this iron. It was a complete accident. So what would happen under this scenario, most likely, is that sensory neurons in the periphery at the level of the hand would detect the painful stimulus and then send messages to terminals in the dorsal root of the spinal cord. This would result in the release of neurotransmitter that would excite the motor neuron in the ventral horn. Axons of the motor horn would then synapse onto the C6 spinal nerve, and this would actually be occurring at the level of the C6 vertebrae. So this would be the 6 cervical vertebrae of the spine. This message wouldn't go all the way to the brain necessarily to get a response. So here you have the axons of the motor neuron joining the C6 spinal nerve and then travel to the biceps themselves, or the biceps brachii, where, and then when terminals of the C6 spinal nerve release the neurotransmitter at the biceps, the muscles of the biceps contract, causing the hand to withdraw. All of this would happen within a split second. It's a very rapid spinal reflex that involves electrical and chemical signal. But what if the painful stimulus wasn't an iron that we touched by accident? What if the painful stimulus was a casserole dish from the oven and you worked like 12 hours on this casserole? You started the night before. The last thing you want to do is drop this casserole. So what would happen here is pain would trigger a spinal reflex, which would make you drop the casserole, but you don't drop it. You get it to the table. What prevented your withdrawal reflex from ruining dinner? The answer is the brain. Top-down control. The brain, or neural circuits within the brain, would be counteracting the withdrawal reflex with inhibitory messages. So what does that look like? So the extra step here would involve the brain, or axons from neurons in the brain, reaching the spinal cord, where the terminals from these top-down inhibitory axons form synapses with inhibitory interneurons in the spinal cord. When activated, the interneuron would then release an inhibitory neurotransmitter, which decreases activity of the motor neuron, blocking or countering the withdrawal reflex. Both withdrawal reflex and its inhibition require neural communication across relatively great distances. But how does this happen, and how would we measure this? So first, as traveling long distances, like the giraffe has the longest axon in all mammals. They have an axon that reaches from the brain down to the base of the leg, and that long axon signal would be repropagated at the nodes of Ranvier, or sort of the gaps in that insulated sheath that coats the axon. But you would measure the electrical potentials of these axons using electrophysiology. Electrophysiology is a technique that would involve an electrode that would conduct, is a conductive medium that can be used to apply electrical stimulation and record electrical currents. We often in neuroscience use microelectrodes, which would be a very fine electrode that's generally used to record activity of individual neurons. While we won't go over this in detail, it's, I think, necessary to introduce the concept of the Hodgkin-Huxley model, at the very least the sign that it's out there. If you go on to enter a graduate program in neuroscience, you will most certainly study Hodgkin-Huxley in depth. But this is a sort of a model that explains neural activity, and it basically has several component parts, but it considers the plasma membrane to be capacitance, and it thinks of this quantitatively, mathematically. The plasma membrane is capacitance. The other things to consider are the sort of the locked ion channels and the leaky ion channels, both of which are taken into account mathematically in their model. And then they measure gradients that result in ion currents. So, but that is all explained using the Hodgkin-Huxley model, which is how you would actually quantitatively study action potentials at the next level rather than at this qualitative level, like what is going on. That would be done using Hodgkin-Huxley. Membrane potential is, an important part of the Hodgkin-Huxley model. This is actually what provides capacitance, and resting membrane potential and the action potential are arguably the two fundamental components here, and we'll break those down individually very shortly. But the membrane potential, which provides capacitance, is the electrical charge across the cell membrane, which is by definition capacitance. This would be the difference in electrical potential inside and outside the cell, essentially a battery. The oscilloscope would be how you'd actually understand or see the readout from the microelectrode. So if the microelectrode were to detect, changes in, current or voltage, and you can either clamp to current or voltage and measure one or the other based on the concept of Ohm's law, the relationship between resistance, voltage, and current. Ohm's law is fundamental for electrophysiology and really part of the basis of Hodgkin and Huxley. But the oscilloscope is how you would actually detect changes here in voltage or current. And it's and it would just display it as a graphical readout for you. So in our next PowerPoint, we're going to go over these concepts in depth. Really just devote the entire thing to the resting membrane potential and action potential. These are two basic electrical properties or potentials of axons. The membrane potential can change, resulting in either a depolarization or a hyperpolarization. Depolarization would be moving or trending toward an action potential occurring. A hyperpolarization would be more breaks, the opposite of an action potential occurring. And all of this could alter the threshold of excitation, or when the action potential is going to fire.